Thank you so much. Здравствуйте. Сегодня мой первый день в университете МГУ. I came here 47 years ago as a student. I loved Russia then, I still love Russia. So I'm back after a long distance, a long delay. Лучше позже, чем никогда. Vice Rector Kakov, Dean Kachuk, distinguished faculty, Adam, Steve, sponsors of the lecture, distinguished faculty, students, friends. It's so nice to be here. I'm going to use this opportunity to talk about the Aquaporin water channels, but I also hope to use this to reignite the issue of international friendships. Science brings peoples together, and as scientific colleagues, we become friends and have a special role. So the title of my talk, Aquaporin Water Channels, From Atomic Structure to Clinical Medicine, and special thanks to Nobel Media, Adam, and AstraZeneca, Steve, for organizing this opportunity. I'm not a very good photographer, but when I was a high school student traveling through Russia, I took some pictures. I've been here before. Here's our group camping. I'm the photographer, I'm not in the picture. We were all teenagers. The gentleman sitting, my high school German teacher, organized it. He was the grand old man. He was 25 years old. We had a fantastic time in Russia. We entered from Finland, Vyborg, to Leningrad, Moscow, South Oryol, Kursk, Kharkov, Pyatigorsk, Ordzhonikidze, all the way to Tbilisi. This was our interest guide, Nadya Mukhina. Nadya, if you're out there anywhere, I'd love to see you again. We camped in interest campgrounds with real Russians, probably the parents, grandparents of the students that are here today. Russia was a little different in 1966. Just 2 decades after the Great War, still recovering. This was the main highway south of Moscow. Where are the cars? There are 2 trucks. There's a motorcycle. It's changed a lot. But the spirit of the Russian people has not changed at all. It's immensely welcoming, encouraging, enthusiastic. Here is our Volkswagen bus. A young couple were so enamored they wanted their photograph taken in front of the Volkswagen Kombi wagon. And we stopped at villages. The villagers all came in to see this wonderful machine, this wonderful Volkswagen bus. Хорошая машина. And this is a picture of me off the Georgian military highway en route from Ordzhonikidze to Tbilisi. It was a great adventure. I'm so glad to be back in your country. So the issue that I'll discuss scientifically is the water channels. As our dean mentioned in the introduction, our bodies are primarily water. 2/3 of our body mass is water. This is shared amongst all life forms. It was Albert Szent-Györgyi who received the Nobel Prize for his work on vitamin C. But he's remembered for having once said, "Water, the solvent of life." Without water, there's no life. For a moment, think now. Our bodies are 2/3 water mass. Is it randomly distributed? And the answer is, of course, no. During this hour while I'm speaking, we'll each be secreting and reabsorbing cerebrospinal fluids to bathe the surface of our brain. We'll be filling the orbits of our eyes with aqueous humor, protecting the surface of our eyes with a thin film of tear. Those of us who've just had something to eat will be salivating. Some of us, me in particular, will be sweating, humidifying our airways, concentrating our urine, a large volume of primary urine, a small volume of final urine. And if you don't think that's important, next time you're on a long airplane flight in the economy section and the seatbelt light goes on, think about Aquaporin 2. It'll make perfect sense. So the issue in terms of biology of water is, how does water cross barriers? How does water cross the plasma membrane? Long before our work started, other biophysicists had considered the problem of transmembrane water permeability. And it was concluded correctly with the discovery of the lipid bilayer, the outer membrane of the cell, flexible, semi-permeable to water, that diffusion will allow water to enter and leave cells. But this was not enough to explain rapid movement of water through renal tubules, through the membranes of secretory glands and red blood cells. It was understood by this pioneering group of biophysicists that these membranes had a very high capacity for water permeation and very selective for water. And the movement was created by osmotic gradients. Osmosis, this process that we all learned about as small school children. Rapid osmosis occurred, but how? So in 1970, the year I began my university studies, an important experiment was done at the University of California, Berkeley, by Bob Macy, a biophysicist, who used a series of chemicals to treat membranes. And what he discovered is that mercuric chloride was a potent inhibitor of red cell membrane water transport. And if he treated the membranes with chemical reducing agents, he could restore water transport. He could turn it off, he could turn it on. And he concluded correctly that the difference between all membranes and water permeable membranes, highly water permeable membranes, is the existence of these putative water channels, the pathway across the membrane. But neither Macy nor others could identify the channel. And in science, theories and hypotheses are fine. Hypotheses in particular are fine. But to become accepted, you must have scientific proof, strong evidence, identify the molecule itself. And it was our laboratory that by sheer serendipity, let me repeat that, we made an observation that led to the identity of the water channel and the Nobel Prize. We were studying the rhesus blood group antigen of red cells. Rh antigens are important in maternal-fetal incompatibility. We identified a new protein of 28 kilodaltons, and in the purifications, a slightly smaller protein co-purified. We cloned out the complementary DNA, and what we found was a series of fragments of genes in the database. One member of the family, MIP, in lens, very abundant but function unknown. Another member of the family, big brain, important in brain development in insects, Drosophila melanogaster, but again, biophysical function unknown. A protein that allowed bacteria such as Escherichia coli to use glycerol as a carbon source, but the actual biophysical function unknown. And finally, a series of fragments from the roots of plants. So we'd identified a new protein, we actually had a putative structure, we had everything but a molecular function. And of course, this is the most important feature of any protein, is what is its molecular function. We were stuck. So how does one make progress when you're stuck? Well, if you're a student, you can talk to your professor. He or she may know, but you could go to the library. The library has a lot of information. It's the conversations with other scientists that oftentimes leads to the answer. And these conversations occur when we least expect important insight. Let me illustrate. So my wife and I have 4 children. These are the center of our lives. They were small, cute little children when this picture was taken 20 years ago. Now they're grown, they're young adults. As a scientist with a modest salary, every year our family vacation was in a tent at the great American national parks. We camped in the Grand Canyon, Yellowstone, Yosemite. And after doing these camping trips several years, Mary said, let's be like a democracy, let's vote. Let's ask the children which national park they wish to camp in next year. And they immediately said, Disney World. Well, Disney World is actually not a national park. But we compromised. We went to the Everglades, we went to Disney World. And on our way back from that trip, we stopped at Chapel Hill, North Carolina, where I spoke with some colleagues. In one of those conversations, when I talked about this new protein in red cell membranes, we knew at that point it's also present in renal tubules, but we didn't have a function. It was a colleague, John Parker, who said, Peter, these are water-permeable tissues. Have you considered this is the long-sought water channel that biophysicists have been searching for a century? Never occurred to me, but that idea, John's idea, caused us to collaborate with Bill Gugino in the physiology department at Johns Hopkins. We did a number of very simple experiments. So these two spherical objects here are the eggs of Xenopus laevis frogs, frog oocytes, about 1 millimeter in diameter. On the left is a control oocyte injected with buffer alone, and on the right is an oocyte injected with complementary RNA for this new protein. RNA leads to synthesis of the protein. If this new protein is a water channel, the one on the right should be osmotically active. We already knew that frog eggs were very, very low water conductivity, so we expected the one on the left not to be osmotically active. So we transferred them from isotonic culture media to distilled water. Kapow! Immediate difference. As you can see, the control oocyte is swollen negligibly, the test oocyte is swollen rapidly and exploded, producing immediate jubilation in the laboratory. Let me be very blunt. If you're a scientist and you make a discovery, and this is not intensely pleasurable, overwhelmingly joyful, you're in the wrong business. This was Greg Preston, the postdoc who did these early studies. I took the photograph 2 years after the discovery. He was still celebrating. So suddenly the problem of rapid osmosis, transmembrane water permeability became clear. It was a new protein which we named Aquaporin, now known as Aquaporin 1, first molecular water channel, basically the plumbing system of the cells. Now, being a small lab, really we were a very small lab. We were not a rich lab, we had a small number of grants. We needed to team up with others, and this is where science is such a rich community. We were able to team up with a number of experts from around the world. I'll show you some of their pictures to do a number of important steps which we could not ourselves do. First, we teamed up with these two gentlemen, Yoshinori Fujiyoshi from Kyoto, Japan, and Andreas Engel from Biozentrum at the University of Basel in Switzerland. They're experts in the technique of membrane crystallography, and in short, we solved this structure. If you look on the left panel, from above the cell, the water channel is a narrow pore through the protein, 3 angstroms in diameter. And I think many of the students will realize the average diameter of a water molecule, 2.8 angstroms, is just big enough. And viewed in cross-section on the right, we have a single channel connecting the outer surface of the cell with the inner surface of the cell, a single aqueous pore, allowing water to move with almost no resistance. And there are barriers to the movement of other solutes. For example, protons, the hydronium ion. They're positively charged domains which repel it. So here we have water in bulk solution, free exchange of protons. Here, water in bulk solution, but this narrow pore, 20 angstroms in diameter, single file movement of water and water alone. And this was work of David Cosino, a talented student in the laboratory who's now a faculty member at Harvard University. So if you look at the space filling model of water at the center of the pore, you'll see it's surrounded by special residues. Arginine, a positive charge. Histidine, a partial positive charge, repelling proton movement. Cysteine, the mercury inhibitory site. Notice it's big enough for water, just big enough. Now, the work of others has extended this structure, and I'll talk about the other homologs in a moment. We were also interested in the physiology of the water channels, as Dean Cockridge mentioned. In order to establish the physiology, we had to know where, specifically at a cellular and subcellular level, where the protein was expressed. And we achieved this by teaming up with Søren Nielsen from the University of Aarhus in Denmark, who localized the Aquaporin 1 protein in the kidney in the proximal nephron. So shaded in green, here's the glomerulus where filtration occurs. Proximal nephron descending thin limb of the loop of Henle. And each of our kidneys has about 1 million nephron units. And the green highlights the location in the proximal nephron where Aquaporin 1 is present. So it's present here, and this is where constitutively high water permeability exists. It's not present in the ascending limb, negligible water transport, and it's not present in the collecting ducts, where regulated water transport was known to occur. Surprising, collecting ducts didn't have it. It was a clear prediction that other homologs must exist in kidney. So 2 microscopic slides. Shown here is a light microscopic preparation of a rat kidney. And what you should see is the tubule here, the lumen where the primary urine flows, is surrounded by an apical membrane, intensely staining with the antibody for Aquaporin 1, present also in the lateral membranes separating the cells and the basal membranes, and not present in the collecting duct. Now, if you look by Immunogold electron microscopy at this apical bush border here, you see infoldings of membrane. I think you can see that. It looks like hair-like projections, and the black dots represent immunogold, nanogold particles attached to an antibody to Aquaporin 1. So the protein is all present at the absorptive surface, not inside the cell, ideally poised for maximum absorption into the epithelium. So how does this allow water transport to occur? The physiologists here will be aware that every day our kidneys will filter about 200 liters of plasma, filtering the same plasma again and again. 200 liters of filtrate is then passing through the convoluted tubules where 99% of the water is reabsorbed. Imagine if you made 99 liters of urine, well, you would last only a few minutes because you would go into shock. So we must efficiently filter and reabsorb the water. But we must reabsorb the water, but not the acid or waste substances. And the explanation is the distribution of the Aquaporin 1. The primary urine flows through the lumen, enters through the apical surface. Tight junctions prevent water from slipping between the cells, exits through the basal membrane. Transcellular movement. Almost 200 liters of water per day. It's amazing. Now, as a medical doctor, I'm very interested in the causes of disease. It's our hope that our scientific work will provide something useful to prevent or treat disease, at least lead to the understanding of disease states. And before the human genome was solved, we localized the chromosome for Aquaporin 1 in the short arm of human chromosome 7, where we had a candidate locus, the Colton blood group antigens. And this is the work of Chol-So Moon, graduate student in the lab. And in short, a long series of experiments, we confirmed that the Colton blood group antigen was a polymorphism in the outer surface of the Aquaporin 1 protein. But we were made aware that in the whole world, only a small number of humans had ever been identified lacking the Colton antigens. Well, obviously, we were very interested in these Colton null people. And because of the wonderful collaboration amongst blood group referencing laboratories, particularly the National Center in Bristol, England, we gained access to a number of Colton null individuals and confirmed that they had mutations in the gene encoding Aquaporin 1. Our thought was, well, this is protein must be so important that they'll be severely disabled. In fact, the Aquaporin 1 null individuals all felt well and looked well. They didn't feel sick at all. Here's a photograph of one. With her permission, we took a picture of a lady from south of France who's a Colton 1 null individual, an Aquaporin 1 null homozygote, with 2 of our colleagues from Johns Hopkins. She looks well and feels well. And most of the time, all of us look well and feel well. But when we're stressed, certain mutations become very clearly manifest. For example, I suffer from a mild form of asthma. If I'm around the laboratory animals, I wheeze. But by using a corticosteroid inhaler and staying away from the lab animals, I'm fine. Others may be sensitive to salt in the diet. Too much salt, they become hypertensive. So silent carrier states are very common. When stressed, we expect this will be a significant phenotype. And Landon King, the young man shown here, did a complete renal tubule analysis of these people and found that they had normal renal function except when thirsted. Every night, if we're lucky, we sleep 7 or 8 hours. We drink no fluid. In the morning, we wake and we've been thirsted 7 or 8 hours. And our morning urine, of course, is the entire nighttime's collection. But the next few drops of urine are highly concentrated. So normal individuals will concentrate the urine from physiologic tonicity, 280 milliosmolar, up to about 1,000 milliosmolar after overnight thirsting. The Aquaporin one null individuals can concentrate a little bit, enough to get through the night, but they would surely get into clinical trouble with dehydration long before any of us would. So it's a pretty important phenotype. Landon, also an outstanding physiologist with interest in lung, demonstrated the presence of the Aquaporin-1 protein in the capillaries of lung, and here's a human capillary. You should see the Immunogold decoration at both the top and the